The exam is done. Happy days are here again, right? How was the exam? I see a lot of nods. I guess that's a good sign. Well, one growl. Yes? So some questions you weren't sure how to answer. Yeah. Okay. Okay. Yeah. Okay. This exam, as you saw, the format was different from the exams last term. And the advantage of the exams this term is I get to ask you more questions. And asking more questions is actually good because it means it's more likely I will find areas that you have expertise in. When you have fewer questions, then it's more likely that you're going to miss things that students have down. So more questions works better in that sense. The exams are not yet graded. They will be finished, I'm pretty sure, by Monday. I will put out a message to everybody letting you know to pick them up. And they will be available in the Biochem office as before, which is ALS 2011. But please don't go there until I send you the email. It won't happen today, I can assure you that. So. All right, so we've got new material to cover, and I've just got a very brief thing to talk about with respect to cholesterol that I didn't finish with last time. It'll just take a couple of minutes to do. And then we'll start talking about fatty acid oxidation, fatty acid synthesis, and some of the things derived from fatty acids as well. So cholesterol, as I've alluded to earlier, is an important precursor of other compounds. So in addition to being important for membranes, cholesterol is also important as a precursor for making what are called bile salts or bile acids, and I use those terms interchangeably. You can see that there's actually more than one step in going from one to the other here. We don't really have to be concerned about that, but these are what you see on the screen, the bile acids. There are several of them. There's only two shown on the screen, and they are acids. So you'll notice there's a carboxyl up here, and here's a sulfate over here. Both of these, of course, at physiological pH have a negative charge, which means that these guys really can act to some extent like detergents. Now, if you recall the structure of cholesterol, it only had one hydroxyl group. These guys have a couple of extra hydroxyl groups, which means that they aren't really completely nonpolar at this end, but they're fairly nonpolar. This structure seems to work pretty well because, as I noted, the bile acids are used in the body to emulsify lipids, okay? Primarily fat in your diet. So the bile acids are in your digestive system, and they help to make those water-insoluble things that we eat, like fat and fat-soluble, fat and, what's the word I want for fat-soluble vitamins, okay? Those aren't normally soluble in water, so we have to, in order to get them into our system, we have to use a detergent to make them be that. And so the bile acids or bile salts accomplish that. The cholesterol is also a precursor of the steroid hormones. And the steroid hormones come in several categories. They're all derived from this compound called pregnenolone, which, as its name sounds like, it comes from pregnant, which it is. Or it isn't pregnant, but it comes from that word. The pregnenolone, as you can see, is a precursor of the progestins, which are used to maintain pregnancy. And the progestins give rise to the glucocorticoids, the mineralocorticoids, and the androgens being the male sex hormones. And you'll notice that the male sex hormones give rise to the female sex hormones known as the estrogens. From a health perspective, this step of going from the androgens to the estrogens is a very important one because it actually is a target for some drugs that I'll describe in just a second. All right, so if we look, we don't need to worry about that. If we look at this guy, what we see is that, well, that's actually not what I wanted either. I thought I had, I don't have it on here. Here it is, yeah. Okay, so if we look at the androgens, such as testosterone up here, we see that they have a structure that, of course, looks very much like cholesterol. And when we compare that to the structure of the estrogens, and by the way, some of the estrogens are shown here on the bottom, when we compare the structure of testosterone to the structure, and androstenedione, by the way, when we compare both of these to the structure of both of these, the most obvious difference that we see is this benzene ring. This benzene ring is made in a rather unusual reaction. In fact, it's a reaction that is not really found anywhere else in biology. This reaction of making directly a benzene ring is a function of an enzyme called aromatase, aromatase. So an aromatase will actually make an unsaturated benzene ring, as you see here. Now, I tell you that because the estrogens are, in fact, hormones that can affect tumors. There are some tumors that have estrogen receptors on them. And there are, I shouldn't say some, there are several that have estrogen receptors on them. And if one can stop the synthesis of estrogen, one can stop or very significantly slow the growth of tumors that respond to estrogen. And so, as a consequence, there are a series of drugs that have been designed that are called aromatase inhibitors. And aromatase inhibitors do exactly what I just said. They stop the conversion of the male sex hormones into the female sex hormones and can be very powerful tools for fighting cancers that have estrogen receptors. Okay? So, very useful piece of molecular knowledge coming from metabolism. Okay. And I think that's, one last thing I want to say is vitamin D. Vitamin D is a steroid vitamin, as it were. It is derived from cholesterol, and it comes from this 7-dehydrocholesterol in a several-step process. You're always told, of course, that if you need to go outside when you're a kid and play in the sun because sun gives you the vitamin D, and this reaction actually shows you why that's the case. So our body can make sufficient quantities of 7-dehydrocholesterol. And if 7-dehydrocholesterol in our skin, for example, is exposed to ultraviolet light that comes in the rays of the sun, it will convert itself to pre-vitamin D. Pre-vitamin D can then be further metabolized to vitamin D3, which is the type of vitamin D that's most commonly found in tablets that you take. And vitamin D3 is converted then to the active form of vitamin D calcitriol, shown here on the left. Okay? So having exposure to ultraviolet light is sort of a yin and a yang thing. The yin part, which one's the good one, yin or yang? But anyway, the good part is that you can make more vitamin D. Vitamin D is very important for healthy bones. It's also important for a healthy immune system. And most people are deficient in vitamin D and they don't know it. Okay? Most people, underline what I said there, most people are deficient in vitamin D and they don't know it. So taking a vitamin D supplement, if there's one supplement you want to think about taking, vitamin D is probably a pretty good one because vitamin D starts right here. So even if you're not getting out in the sun and so forth, the compound that's contained in the tablets you take is here. You don't need sun to activate that. So that's a very important consideration. But as I said, these are all, again, derived from cholesterol. You can see this guy up here looks very much like cholesterol, and that's where its origins come from. Okay. Any questions about what I've just said? You guys are ready to move on? Yes? Do I know how light excites the reaction? Yeah. Okay. So it's actually what it's doing is the light causes this electronic rearrangement that you see up here on the screen, basically breaking that bond of that ring that's there. That's what light is doing. Yes?
Regarding the tumors
yes.
that you're treating,
Mm-hmm.
doesn't that affect the body in other ways?
So does treating the body with aromatase inhibitors affect the body? Yeah, you bet. So you may go into menopause, et cetera, as a consequence of that, but it's better than having a tumor that's growing. So, yes. Okay, let us move on and talk now about fatty acid metabolism. Fatty acid metabolism is actually rather straightforward to learn because the chemical process is very similar in the forwards direction compared to the backwards direction, the backwards being chemically very much like the reverse of the forward process. Now, there are some considerations for it, and the backwards process, that is the synthesis of fatty acids, does require additional energy, and more energy than what fatty acid oxidation gives us, but nonetheless, chemically, they're very, very similar to each other. In our body, and by the way, I find students frequently confuse fat and fatty acids. So fatty acid is a component of a fat, all right? Fatty acids, as I told you earlier, in the body move by being carried by serum albumin, and fats in the body are stored in specialized cells called adipocytes. There is an adipocyte that is very happy, okay? Very, very happy. I have too many of those adipocytes. I should have them less happy. This figure is nice because it shows us the chemical relationship between fatty acid oxidation and fatty acid synthesis. Now, there's one other step of fatty acid synthesis that's not shown on here, and I will tell you more about that later. But suffice it to say, if we look at the fatty acid degradation, which of course is oxidation, we see that it proceeds in 4 steps. Okay? So it's an oxidation, a hydration, meaning addition of water. There's another oxidation, and then there's cleavage. So those are the 4 steps. And if we look at the steps going in the reverse direction, instead of cleavage, we have condensation, which is the opposite of cleavage. Instead of oxidation, we have reduction. Instead of hydration, we have dehydration. And instead of oxidation, we have reduction. So the processes are very, very similar in opposite directions chemically. Okay? They are occurring in different places. So fatty acid oxidation is occurring inside of the mitochondrion, for the most part. We'll see that there is also fatty acid oxidation that occurs in a specialized organelle called the peroxisome. Fatty acid synthesis, for the most part, occurs in the cytoplasm. We will see that there is also a little bit of fatty acid synthesis that occurs in the endoplasmic reticulum. There's also, interestingly, a little bit of fatty acid synthesis that occurs in the mitochondrion. And we'll see how the cell manages that a little bit later. Okay. All right. So these guys then are sequestered for the most part. They're sequestered, oxidation occurring one place, synthesis occurring in another place, with the exception of that little piece I'll describe later in the mitochondrion for synthesis. Okay. There's another bile acid. Up close. Question? Oh, okay. Another bile acid that I'll remind you again is used to solubilize fat. Here we look a little bit more closely at the breakdown of that fat that occurs in our body. And one of the things I didn't talk about when I talked about the movement of fats and fat-soluble vitamins through our digestive system is actually what you see on the screen here. Okay, it's kind of interesting. If we think about eating that McDonald's cheeseburger that's full of all that fat and so forth, in order to get that fat across our digestive wall, our digestive system, there's a couple things that have to happen. And they actually involve breaking down partly the triacylglycerol into a monoacylglycerol. Okay? Now, this involves the action of enzymes called lipases. So you can see one lipase cuts the triacylglycerol, cutting off a fatty acid and leaving a diacylglycerol. And that diacylglycerol can be broken, another fatty acid can be taken off of it by another lipase, leaving us with a monoacylglycerol. Okay? These guys, the fatty acids and the monoacylglycerol, are then moved across the digestive system. And then, on the other side, they're reassembled. That seems kind of odd, right? They're taken off, they're moved across the digestive system, and then they're reassembled back into a triacylglycerol so they can go into the chylomicrons. Why does that happen? Well, I don't have a real good answer for it, but my best answer for it is that if we look at these guys, this is totally insoluble in water. And these guys, the fatty acids that are cleaved off, act a little bit like detergents. So remember, we've got bile acids in our digestive system. It may be that these guys help to emulsify things as the transport occurs and as the chylomicron is being made. So that may be part of the reason why these are taken off and then put back on. There's not a real good answer for why that occurs, though. Okay. Yes? So the so what's moving across is this guy and the two fatty acids, right? So we no longer have a fat once we've taken the fatty acids off of it, right? Okay. Okay. Let's see. So here we are. We see it. There's our fat. There's our fatty acids. There's our monoacylglycerol. They come back over here. They're made back into triacylglycerols and then assembled into chylomicrons, which then go into the lymph system. Okay. Breakdown of fat within cells. Now, what I've just described to you is what's happening in the digestive system. Okay. If we think about the breakdown of fat that we have stored, we have to think about breaking down what's in the adipocytes. Now, this is something that we really want to do in a diet, right? Because in a diet, we want to reduce our adipocytes. We want to burn that fat. So what's happening here is a very similar thing. Okay, triacylglycerols are being broken down. In this case, all three are clipped off, and we're left with 3 fatty acids and a glycerol. The very first enzyme that cuts the very first fatty acid off is regulated by a hormone. Okay? Now, this turns out to be significant because this is the only regulation of the breakdown of fat. The only regulation. This hormone affects, the enzyme is called triacylglycerol lipase. And look, here's our friend, a 7-TM receptor. Here's a hormone binding. Here's a G protein interacting with adenylate cyclase. You've already seen all this stuff before, right? Releasing cyclic AMP, activating protein kinase A. And what is protein kinase A doing? Well, it's affecting several of these enzymes here, but the one that's of interest to us is the triacylglycerol lipase, okay? So that's this guy right here. This guy is becoming activated here. You don't have to memorize all these other things in there. The important thing is that triacylglycerol lipase is activated by this hormone system. That regulation is, as I said, the only regulation of fat breakdown in our bodies. Okay? Now, we're interested in talking right now about the metabolism of those fatty acids. So at this point, we have fatty acids. The fatty acids have been broken down by the fat, and we remember that when the chylomicrons and so forth go transported through the bloodstream and so forth, the cells can release lipases to pull up fatty acids from them. They can also pull fatty acids out of the serum albumin. So these guys that were broken down in the adipocytes are most definitely going to hit the serum albumin, and they're going to get carried to target cells, which are going to take those fatty acids. Okay? All right. Now, I'll show you one trivial thing, and then we'll talk more about oxidation. The trivial thing is that glycerol, which is the other component of fat, can also be metabolized by us. And it can either be oxidized in glycolysis reactions, so we can see the conversion into a glycolysis intermediate here. Here's glycerol becoming glycerol 3-phosphate, becoming DHAP. Well, DHAP, of course, is a glycolysis intermediate. At this point, DHAP can either be oxidized to make pyruvate, or it can be converted into glucose in gluconeogenesis. So depending upon which direction the cell needs things, it can go either direction. Now, I mention this to you only because this is the only part of fat that can be made directly into glucose. It's the only part of fat that can be made directly into glucose. And even it isn't always. But glycerol is the only part of fat that can have that happen. The reason fatty acids can't be made into glucose is because we don't have a glyoxylate cycle, as we talked about last time. Those fatty acids, as you will see, get broken down into acetyl-CoA. And acetyl-CoA cannot be made in animals into glucose. And let's see, we'll let that be. Okay. So let's talk now about fatty acid oxidation a little bit more closely. Cells have to handle fatty acids. We've talked before about the fact that fatty acids are problematic for cells because they can act like detergents, and detergents can, of course, denature proteins. So if I have a cell that has taken up a fatty acid from serum albumin, all right, the cell really needs to do something with it fairly quickly because it doesn't want that fatty acid denaturing proteins and causing some other problems. So what the cell does is it takes that fatty acid, links it to coenzyme A, and makes what we call an acyl-CoA. That's ACYL. ACYL refers to a carbon-hydrogen chain, in this case with an acid at the end. This acyl-CoA, okay, is just like acetyl-CoA. Acetyl-CoA has 2 carbons. This guy might have 16 to 24 carbons in it. We can see that the addition of this CoA is energetically expensive. ATP is converted into AMP, and that means that's the equivalent of 2 ATPs that are being used to make this thing. So it's the equivalent of 2 ATPs. So this is an important thing that the cell's doing. It's not doing it just for random purposes. Then the cell does something rather odd. Okay? The odd thing it does is it takes that acyl-CoA, which we see right here, down to the mitochondrion, because fatty acids are going to be oxidized inside of the mitochondrion. But oddly enough, the cell does not have a transport mechanism to move the acyl-CoA across the mitochondrial inner membrane. Okay? So the cell has just wasted 2 ATPs, putting a CoA on there, and then it can't use the acyl-CoA to move across the membrane. So what does it do? Well, it takes that CoA and it replaces the CoA with another carrier called carnitine. Carnitine. And the cell does have a transport protein that will move an acylcarnitine across the mitochondrial inner membrane. This is the mitochondrial inner membrane here. Okay? So we see the acylcarnitine coming in, and once it gets into the matrix of the mitochondrion, which is where the reactions will occur, once it gets into the matrix, the carnitine is taken off and replaced with a CoA. This is a very odd system. So the cell has taken in an acyl group, a fatty acid. It's put a CoA on it. It goes to the mitochondrion, takes the CoA off, replaces it with carnitine, moves it across the membrane, and replaces the carnitine with CoA. Okay? So there's a shuttle that's getting us in here. Now what we have in the mitochondrial matrix at that point is acyl-CoA. Okay, so hopefully I haven't confused you too much. Yes? This is the mitochondrial inner membrane right here. That's right.
Why wouldn't the cell put the carnitine on it in the first place?
So why doesn't the cell put the carnitine on it in the first place? That's a very good question, because it doesn't. Yeah, I don't have an answer. I mean, you would think that it's investing this energy in doing this thing. Why doesn't it make that? I don't know the answer to that question. Yeah. Yeah.
Would there be not enough energy to put the acyl onto the CoA?
That's a good question. So is there enough energy that's put into this to make drive the reaction of carnitine? I would say probably not, because down here we see the reverse of the reaction. So it's a good thought, but I don't think, because then we would have to put energy into this to make that happen, and it doesn't look like we do that. So I don't think there's a big energy difference between the acylcarnitine and the acyl-CoA. If we think about the history of mitochondria, mitochondria were originally, cells, free-living cells on their own, and they had their own mechanisms for doing things. Maybe what they had was something besides a CoA that got carried across, and the cell says, okay, well, we'll adapt to that. Now, why the cell didn't put carnitine on it to begin with, I don't know. Yeah? Well, I won't say they're wasted, because this carrier on there is important. Okay, so maybe if I may have used that word. If I said that word, wasted probably isn't the right word to use. It's an energetically expensive thing that they're going through. Covering up that fatty acid carboxyl end is important. Okay, so whether we put a carnitine on there, or a CoA on there, or a Kevin Ahern on there, it's important to block that off. So I don't want to say it's completely wasted. No. Okay. So we've now gotten into the mitochondrial matrix, and we can start the oxidation process. Now this is very much like what you saw in that figure that I showed you earlier. I'm going to step you through it step by step a little bit more carefully and tell you what's going on. There are several enzymes. In fact, there are 4 enzymes that are involved in this process, and I only expect you to know 2 of them by name. Okay? You should know the reactions. It's important to know the reactions. The enzymes number 2 and 3 have names that are about this long, and I don't see them memorizing those names are particularly informative to you. The first one, however, is a very important enzyme. Okay? So this enzyme is acyl-CoA dehydrogenase. So a dehydrogenase, we remember, always tells us that we have an oxidation going on. And this acyl-CoA dehydrogenase pulls out protons and electrons between carbons number 2 and 3. In fact, we're going to see in all of fatty acid synthesis and degradation that all the action is between carbons 2 and 3. The only exception to that is the oxidation of some of the unsaturated fatty acids. But with that exception, everything occurs between carbons number 2 and 3. So we can see here's carbons number 1, 2, 3. We see 2 and 3 are already shown here in red. The oxidation reaction is pulling those protons and electrons off. They have to go somewhere, and they get put onto FAD to make FADH2. What reaction that we've seen previously takes electrons and protons and puts them onto FAD to make FADH2? Anybody remember the enzyme? Succinate dehydrogenase. There was another dehydrogenase, and that was in the citric acid cycle. In that case, if you go back and you look at it, you'll see that electrons and protons get pulled out between carbons number 2 and 3 there also. Okay? All right, so we have this intermediate now that has a trans double bond, and in the citric acid cycle we make fumarate that has a trans double bond, so everything is very similar to that. The next thing we do is we add water to that. That's a hydration reaction that you can see right here. The hydration reaction converts this guy down here with a double bond to this guy down here that has an OH on carbon number 3. Okay? The same thing happens in the citric acid cycle, where we take fumarate and we add water to it, and we make malate. Now we can either number malate with the hydroxylate number 2 if we put it at the top, or number 3 if we put it at the bottom. So again, the designation is exactly the same. It's going into the same place. It's the same kind of a reaction that's occurring here. Okay. The next reaction that occurs is the oxidation of that hydroxyl that we see. And that oxidation involves transfer of electrons and proton to NAD to make NADH. And it produces this compound here, which is this 3-keto-acyl-CoA. By the way, I don't care that you even know the names of these guys. I think that that's just not really necessary. But you should know the general structures and so forth that we're talking about. Okay. At this point, we are one step away from producing acetyl-CoA, and that step occurs in the last reaction called a thiolysis, and that's another enzyme name that you need to know. This enzyme is called thiolase, Thiolase. So what we've done at this point is we've started with a fatty acid that had a certain number of carbons. And by the way, most fatty acids start out with an even number of carbons. And we've produced a fatty acid that has 2 fewer carbons. And we've produced an acetyl-CoA. Now, this remainder, the acyl-CoA that's shortened by 2 carbons, can then get oxidized in the next step, just repeating this process again. So this will become the starting point up here, and we go around, and we go around, and we go around. The most common fatty acid that we have in our body is known as palmitic acid. It has 16 carbons. We'll talk about some other ones. But if I have 16 carbons, I do this cycle 7 times, because the last one splits 2 in half. Right? I do the cycle 7 times, and I get 8 acetyl-CoAs. Now, this cycle has medical relevance, And the medical relevance is that first enzyme that I described to you, the acyl-CoA dehydrogenase, comes in 3 different forms. A form that works primarily on long fatty acids, long chain, large numbers of carbons. Anything longer than about, let's say 18 or so, long chain fatty acids. Those long chain fatty acids start their oxidation not in the mitochondrion, but they start in the peroxisome. And that's where you see the long chain acyl-CoA dehydrogenase. Now, we'll talk about peroxisomes later, but I just want you to plant that thought in your head. So the long guys start out there. Then they get transported to the mitochondrion, where they encounter the medium chain acyl-CoA dehydrogenase. And this is the one that has the medical relevance. Okay? Infants that die of sudden infant death syndrome sometimes, not always, but sometimes are deficient in this enzyme. So infants with sudden infant death syndrome sometimes are deficient in the medium chain acyl-CoA dehydrogenase. It's thought that perhaps they're unable to metabolize fat and fatty acids that they're getting from mother's milk appropriately, and that leads to their death. Now, it doesn't happen in every case, so it's not a cause-effect, but there is a correlation. All right. In the mitochondrion, there's another acyl-CoA dehydrogenase, and you can probably guess from what I've told you so far, it's going to be called the short chain, and it is. Okay? So it's going to oxidize things down from about 8 or smaller, 8 to 10 or smaller. Okay. Well, at this point, we have oxidized our fatty acid. That's pretty cool. We've oxidized our fatty acid. This shows the oxidation. Let's see. Blah, blah, it's the same thing. This shows the oxidation, then, of the saturated fatty acids. Saturated fatty acids. And by the way, this pathway I just showed you has another name. It's called beta oxidation. And I'll show you why we say that. Okay, beta oxidation. If we look at the molecule here, this bond here, this first bond between the carbon and carbon is known as the alpha. Actually, the first carbon is known as the alpha. The second carbon is known as the beta. So beta oxidation is occurring right here at this point. And that's where the beta oxidation gets its name. Okay. The summary of fatty acid oxidation, there's all those long mouthful of enzyme names. All right. I didn't ask you to know the name of the enzyme that put the carnitine on there either, because it's also got a long mouthful of a name. Okay. But acyl-CoA dehydrogenase, which are right there, several different isozymes, that's an important one. And the other one is thiolase. You see here it's called beta-ketothiolase. You can just call it thiolase from my perspective if you want. And there's a reason for learning that one, and as we will see, that enzyme catalyzes a reversal of the reaction that I've already shown you, and that reversal leads to the synthesis of ketone bodies. So that enzyme plays a dual role, and that's why I want you to know the name of that enzyme. Okay. Now, if we oxidize fatty acids, or if we have fatty acids that are unsaturated, it turns out that there's some more considerations for how we metabolize them. Let's think about this. When I showed you the fatty acid oxidation for beta oxidation, I showed you that there was an intermediate in there that had a trans double bond. And we hear a lot about trans fat, for example. Is this relating anything to trans fat? All right? It's not. So the trans bonds that we're making here occur quite naturally. They have a very specific structure. That trans bond is located between carbons number 2 and 3 on the pathway to the breakdown of fatty acids. Trans fats, by contrast, are made chemically by partially hydrogenating vegetable oil. Now, what happens when you partially hydrogenate vegetable oil? First of all, why do you partially hydrogenate vegetable oil? People do that so they can lower the melting point of the vegetable oil. And if they want to make cookies, for example, they want things to be nice and solid when they're incorporated into the cookie. Okay? So they do that. Well, it turns out that when you partially hydrogenate, if you fully hydrogenate you would have a saturated fatty acid because you would remove all the double bonds. So partial hydrogenation is leaving some of the double bonds behind, and it turns out that during that partial hydrogenation, some of the cis bonds, which is what we normally find in fatty acids, are converted to trans bonds. That's giving rise to trans fat. Trans fat has some very bad things associated with it. They cause inflammation in your arteries. They're linked to things like obesity and also linked to things like atherosclerosis. My guideline to students is go home, take a look at anything, any of your food that's in a box, and look on the back of it and see if you see the words partially hydrogenated. Okay? If you see the words partially hydrogenated, you are eating trans fat, even if the front says contains no trans fat. How can that be? Well, that can be because the food industry has lobbied to get a rule that says if you have less than 1 gram of something, it's the same as none. Yeah, okay? So if it says partially hydrogenated anything, vegetable oil, coconut oil, you name it, if it has the words partially hydrogenating, you're eating trans fat even if you thought you weren't. Okay? Avoid trans fat. Now, I can't tell you exactly why trans fat is problematic. It's one of the first questions I'm going to get. I can show you how trans might be a problem in the metabolism of fatty acids, but I can't tell you that's why trans fat causes the problems that it does. Okay? I would like to be able to tell you, but I can't. So I do want to show you how the double bonds come into play in our body. Okay? So we have plenty of unsaturated fatty acids in our body. Our cell membranes use unsaturated fatty acids. You've seen that, giving some fluidity and so forth. So those, when we break them down, obviously we want to, and in our diet as well, we're eating oils and things like that, vegetable oil, we are getting some unsaturated fatty acids in the fat that we're eating. So how do we break those down? Well, here's a fatty acid called palmitoyl-CoA. It has 16 carbons, and it has a cis double bond. Remember that fatty acids that are made normally almost always are in the cis configuration. There's only a few places where we get trans fatty acids that are actually made. So cis fatty acids is by far the most common. We can see the cis bond here, and we can see that this guy is going to start out and it's going to go through several rounds of beta oxidation until we get to a point where the cell can't go any further. That is, the enzymes that would break this down can't deal with this. Why can't it deal with this? Well, let's look where all the actions are occurring. Here's carbon number 1, 2, 3, and 4. So between carbons 3 and 4 here, we have a cis double bond. This enzyme, which normally would be working right here between carbons 2 and 3, won't touch it because this bond at between 3 and 4 is affecting it, and it won't work. So if we are going to metabolize this, we have to convert this into a form that our cells can use. Well, it turns out we have an enzyme called I like to just call it enoyl-CoA isomerase. You can call it the last half of that name, enoyl-CoA isomerase. And it does something very useful and something very simple. Very useful and very simple. It converts a cis bond between carbons 3 and 4 into a trans bond between carbons 2 and 3. But we just made a trans fatty acid. Yeah, but this is a normal intermediate in the breakdown of fatty acids. The next step would involve addition of water, just like a normal beta oxidation, and then thiolytic cleavage. This will be metabolized just fine. What if we had a trans double bond up here because we've been eating a lot of trans fat? Will this trans double bond be handled by this enzyme? The answer is probably not. So we could see this might gum up our machinery. If the trans double bond is in the wrong place, instead of a cis bond, we may have some problems, Houston, and in fact, that may be one of the places where we can imagine that trans fat could cause us a problem. Okay? Okay. So enoyl-CoA isomerase is a very important enzyme. Not all fatty acids come nice and simple like I just showed you. Some are polyunsaturated. And the positioning of their double bond varies. The positioning of their double bond varies. Now there's actually an error on this figure, and I'm going to try to step you guys through it, if I can remember it right myself. Here's a fatty acid called linoleoyl, what a name, CoA. It's derived from linoleic acid, and it has two double bonds in it. They're both in the cis configuration, as you can see. We go through normal fatty acid oxidation, and we get down to the place that we were in the previous one. That is, we have a double bond that's in a cis configuration between carbons 3 and 4. Enoyl-CoA will handle that one just fine. It handles it just fine and puts that cis bond between 3 and 4 into a trans bond between 2 and 3. And the cell says, fine and dandy, let's go ahead, add water to that, let's go ahead and oxidize that, and let's break off the carbons, and we're a little closer to our target, which is over here. So this, going from here to here, is several steps, and now we see we've got another double bond we have to deal with. This double bond is not between 3 and 4, but it's instead between 4 and 5. Well, you know chemistry, you know enzymes, and you know that position is very important. The first thing that the cell does is it breaks off two of those. The first thing that the cell does is it starts the oxidation process. So it says, okay, well, here's carbons 2 and 3. The first enzyme, the acyl-CoA dehydrogenase, says, no problem. I'll pull off those electrons and protons. So we make a trans double bond right here, just like we would in regular fatty acid oxidation. All right? Then we get a curveball, okay? We can't, and the enzymes that would normally add water and then go ahead and oxidize this can't do it. So instead of metabolizing this, there's another enzyme that comes into play called 2-dienoyl-CoA reductase. Yes, that name is probably important. Okay? It does something interesting. It uses some electrons and protons. So that tells us we started with 2 double bonds. We're going to have either 1 or 0 when this guy is done because it is a reductase. Okay? This reductase now converts this guy here up to this guy, with the exception of the fact that your book has the figure drawn wrong. It draws it as if it is a trans double bond between 3 and 4, and I've just told you that enoyl-CoA isomerase works on a cis bond between 3 and 4. This should be in the cis configuration. This enzyme is making a cis-configured double bond, not a trans. Okay? Well, that turns out to be really useful because now we've got a structure just like we had over here. A cis between 3 and 4 goes to a trans between 2 and 3, and we're set. Now, that's a lot of 2s and 3s and numbers and blah, blah, blah. It takes more words to describe it than it takes to sort of lay out what's actually happening here. Okay. Because of these two enzymes, you notice we've only used 2 different enzymes from before. Because of these 2 enzymes, cells can handle any regular unsaturated fatty acid that they encounter. Any regular unsaturated fatty acid that they encounter can be handled thanks to the action of these 2 enzymes. Again, you could imagine if you have trans double bonds in positions that you wouldn't normally, these enzymes wouldn't encounter, we may gum up the process, and that may be why there's a problem with metabolizing trans fat. Okay, I'll stop there. I've gone through a lot of numbers there and ask for any questions that you might have. You guys are a quiet group today. Yes? Okay. Where did the NADPH come from? NADPH is actually made in the cell as a product of several things, the most common of which is something called the pentose phosphate pathway. So the pentose phosphate pathway has several primary functions, one of which is the production of NADPH. We're going to see that NADPH, and it's very good that you noticed that, NADPH is used in the synthesis of fatty acids as well. So there's in the cell a sort of a dividing line. It's not absolute, but there's a sort of a dividing line. NAD is used primarily for oxidation type reactions. NAD becomes NADH. You've seen this happen many times, okay? NADPH, on the other hand, is used for synthetic reactions. So when we go to make fatty acids, NADPH will be used, not NADH. Okay? So cells have, and they have, as I said, it's not a rigid dividing line. There's nothing being synthesized here. There's not a rigid dividing line, but that's the general tendency, is that biosynthetic reactions tend to require NADPH, not NADH. Okay? Thanks for asking. Yes? So I want you to understand what's happening with these enzymes. That's correct. Am I going to ask you to redraw this figure? I'm not going to ask you to redraw this figure. Because for one thing, if you know what this enzyme does, you know most of it already. If you know what this enzyme does, you know it. Right? This enzyme is the more complicated of the two. All right? But you can see, here's a substrate, here's a product. Here's a substrate, here's a product. That's the gist of what's happening on this figure. Okay? Yes?
I don't understand what happens to the molecule after the enol-CoA isomerase converts this bond.
Oh, so what happens after you get to this point?
Yeah.
Okay. Beta oxidation. So beta oxidation, remember this is an intermediate in beta oxidation. This is not an intermediate in beta oxidation, right? Because you can't handle this enzymes of beta oxidation can't handle that double bond. This is making that double bond such that the enzymes of beta oxidation can handle that. So you would add water, you would oxidize, you would go through thylated cleavage, and you would end up with this. Does that make sense? Yeah. I wish they'd put those other things on here, because then you would see more of what happens. That's a common question. Is magic happens or what? Magic doesn't happen too often. Okay, so that's fatty acid beta oxidation. I'll show you a couple of just quick considerations. One is propionyl-CoA. Propionyl-CoA has 3 carbons. Now, I told you that most fatty acids in the body have an even number. So if I start with 16, and I cut down 2, cut 2, cut 2, cut 2, I get to a place where I have 4, and I cut the 4 in half, and I'm left with everything as acetyl-CoA. Well, if I had something to start with 15, and I keep cutting, 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 I will get not to a point of 4, but I will get to a point of 3. That's propionyl-CoA. Cells have to have a way to handle that, and they do have a way to handle it. It's a very odd reaction that I'll say more about next time, involving vitamin B12. I'm just going to very briefly tell you here, and then we're going to sing a song. All right? The very odd thing that happens is this 3-carbon intermediate is converted into a 4-carbon intermediate. Simple enough. And then the cell decides that it didn't like the place it put that carbon on, so it moves it to a different place. It's kind of like why did you put CoA on, then you took it off and put carnitine on. And then, after it puts it in the new place, it says, oh, I really meant to put it at the end. 3 steps. Okay? Very odd steps. But the product of that is propionyl-CoA has been converted into succinyl-CoA. And of course, succinyl-CoA can be used in the citric acid cycle. So we don't accumulate propionyl-CoA as a consequence of this. Okay, I'll say a little bit more about that at the beginning of next time. Let's finish with a song. This is a song that's very relevant. It's called When Acids Get Oxidized. The fatty acids carried by CoA, CoA, are oxidized inside the mitochondriae. They get to there, as you have seen, by hitching rides on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true. It's true. They jump from complex 1 onto CoQ, CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex V, you see. They do this to make lots of ATP, ATP. The mechanism you should know goes through the stages LTO. So there's energy when Acids get oxidized. See you guys on Monday.